Hello.
Hello. Good morning, sir.
Good morning, ಶಾಸ್ತ್ರಿಗಳೇ. ನನ್ನಲ್ಲಿ ಇಲ್ಲಿ ಇಂಟರ್ನೆಟ್ ಸ್ವಲ್ಪ ಕನೆಕ್ಟಿವಿಟಿ ಇದ್ದಿದ್ರೆ ನಾನು ಮೊಬೈಲ್ ಅಲ್ಲಿ ಜಾಯ್ನ್ ಆಗ್ತೇನೆ. ಐ ಆಮ್ ಟ್ರೈಂಗ್ ವಿತ್ ದ ಲ್ಯಾಪ್ಟಾಪ್. Unfortunately, this room internet connectivity is poor.
ನನ್ನ ಕ್ಯಾಮೆರಾ ಆಕ್ಟಿವೇಟ್ ಆಗ್ತಾ ಇಲ್ಲ, ನೋಡ್ತೇನೆ ಸರ್ ನಾನು.
ಸರಿ.
My camera is not getting on. I'll just find out somebody's help.
ಆಯ್ತು.
So good morning, sir. Can I go ahead?
Morning.
Nimmi.
Yes, sir.
My camera is not getting on, and with your permission, I think you can hear me.
Yes, sir.
I will proceed with the things, and I'll ask some Ramya or somebody to set my camera on. Maybe I'll go off, but initially we'll start.
Yes, sir.
Nimmi, you are muted. Voice is not audible.
How to go?
Yeah.
Start, sir. A very good morning to all. I, Dr. Nimmi Varghese, Assistant Professor, Department of Pharmacology, and my team are delighted to welcome you all to the national webinar on drug discovery for neurological disorders. It gives me immense pleasure in welcoming our Honorable Vice Chancellor, Dr. Satish Kumar Bandari, our Principal, Dr. C.S. Shastri, the Speaker of the Day, Dr. R.V. Om Kumar, and all the delegates. Today's event is organized by the Department of Pharmacology, NGSM Institute of Pharmaceutical Sciences, Mangalore. The topic for today's talk is multi-target directed ligands for neurological disorders. And our resource person is eminent scientist Dr. R.V. Om Kumar, head of the Molecular Neurobiology Group, Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram, Kerala. I pray that the grace and favor of the Almighty God be with us throughout the program so that we may have a seamless webinar experience. Now, I request Principal Dr. C.S. Shastri, NGSM Institute of Pharmaceutical Sciences, to deliver the welcome address.
Thank you very much, Dr. Nimmi. I think there is some sort of echoing going on, and I request all the other participants to mute their devices, possibly. Yeah. Very good morning to all of you, and I congratulate Nimmi for bringing Dr. R.V. Om Kumar. Very good morning, Professor Om Kumar. This is first time we are meeting. Later on, I will show my face once my glitches with the camera is sorted out. In between, I will come and show my face. I am Dr. Shastri, the Principal of Pharmacy College. I am sure that Nimmi must have given a briefing. Sir, we have got the presence of our Honorable Vice Chancellor, Dr. Satish Kumar Bandari of Nitte deemed to be university. Our university is a very comparatively young university. We are around 15 years old now. And we have got dental, medical, pharmacy, nursing, and engineering schools under the university. And we have ranked 74th and in NIRF. And also we have the pharmacy college is ranked 45th in All India ranking framework. So obviously a college of such standing will have various areas where the students are working, and our research scholars are also working. About 40 people are pursuing PhD at this point of time, and some of them are interestingly working in the area of in which you have got a lot of prominence, and you have worked for a long period of time. That is what you call neurological disorders and neurobiology. Disease, all of us know that, which is very difficult to handle, and no obvious remedies as of now, whether it is Alzheimer's or PD or AD or any of these things, we are not able to arrive at a solution because of the obvious complexity involved in the neuroregeneration or restoration of neuronal functions and so on and so forth. Sir, in NGSMIPS, we are focusing on working on some of the plant-based remedies for the management of Alzheimer's disease as well as Parkinson's disorders. Now, of course, we have to take it to the second level because we have been, we have been working on isolation and testing it on animal models, but so far we are not able to take it for the clinical trials. That is where we intend to go in the days to come. And I am extremely glad and honored because of your presence and association with the Nitte deemed to be university as well as NGSMIPS, because you have been working on this neurobiology division of Rajiv Gandhi Center for Biotechnology in Thiruvananthapuram. I am really happy that Nimi could bring you together to the group of Nitte group, and in the days to come, we can have several collaborations, and certainly some of my people, the people who are doing in this area, I'm sure that you will be able to help them and handhold them, and together we can do a lot of work. And I also take this opportunity to invite you physically, not virtually. Of course, the COVID has to permit us to join, but sooner or later it will happen because the intensity or the virulence is coming down slowly. And I'm looking forward to meeting you in person, sir, and coming and guiding my students, postgraduates or PhD students, as well as some of my faculty who are working in this area. Sir, we are honored by your presence, and that is in a nutshell. And I'm sure that our Vice Chancellor will have some words. He will have some interactions, and we can take this particular initiative forward. I sincerely believe and wish that it should not be a one-time interaction with a lecture by you. We have to be in touch and take the things forward. And a person of your eminence will be of great help and inspiration for all of us. With this brief introductory remark, I congratulate Nimi for bringing you and coming us together. And that's all what I want to say at this point of time. In future, I'll be communicating with you over mail or in telephone, and let us work this together, sir. Welcome to Nitte Group, sir. Very warm welcome. And over to you, Nimi.
Thank you, sir. Now I request our Vice Chancellor, Nitte DU, Dr. Satish Kumar Bhandari, to kindly deliver the opening address.
Very good morning. Am I audible?
Yes, sir.
Yeah. Because the connectivity is poor here, I'm connecting through my mobile. Anyway, very good morning. Very distinguished speaker for this very unique program. Unique in the sense, the subject which our pharmaceutical institute has selected is very, very unique. The drug discovery for neurological disorder. And the guest is also very special, as Professor Shastri said. Dr. Om Kumar from the Center for Biotechnology from Trivandrum, Kerala, is joining us in this webinar. Very distinguished speaker, Dr. Om Kumar. Welcome to Nitte University. Even though it's not physical, you are, as Shastri said, you will be visiting us in the future. Welcome you to this webinar on my personal behalf. Professor Shastri, then Dr. Guru Raja, the head of the Department of Pharmacology and the coordinator for this webinar, Dr. Nimi Varghese, and Vice Principal Narayana Charilu, and the faculty of NGSM Institute of Pharmaceutical Sciences, and my dear postgraduate students, PhDs and the students. It's my privilege to be part of this webinar. In spite of my personal difficulties, I am joining you for this meeting because of the great respect I got for the Institute of Pharmaceutical Sciences. NGSM Pharmaceutical Institute of Sciences is one of the oldest constant college of Nitte Education Trust, and it's one of the most active constant college, doing a lot of work, not only academics, also in research. And coming to this topic of drug discovery for neurological disorders, as Shastri said, is one of the very important aspect of the disease burden which we are facing. Is not only globally, the country, the estimation of neurological disease burden is increasing dramatically. The last, I understand, the last 4 or 5 years, the number has almost doubled. As you know, one of the important reason is the life expectancy of the people is increasing in our country, and naturally the neurological disorder, particularly the non-communicable and degenerative and particularly the traumatic neurological disorders are increasing. As Shastri said, the pharmaceutical interventions in neurological disorders is not all that easy, because most of the diseases, the etiology, the pathogenesis itself is ill understood when you talk about non-communicable diseases of the brain, particularly the degenerative disorders, or it may be dementia, it may be Alzheimer's disease, it may be stroke, or Parkinsonism or epilepsy. The management has become very complicated, but still we have to work, and I think it's an endeavor for all of us, particularly young colleagues, researchers, look at the new drugs which can effectively target these cells and nerves, neurons, so that we can conquer this disease, very debilitating disease, so that it will help our common man and the mankind. With that intention, I think Dr. Nmda Verghese, if their very proactive intervention, she has called very distinguished speaker, Dr. R.V. Omkumar. I understand you got vast experience in particularly the neurological disorders, the drug discovery, and you definitely will be of great inspiration and source of help to the faculty and students of our institution. I once again invite you to join us when the things improve in the campus. I think you should handhold us in particularly developing drugs or molecules in the area of neurological disorders. I congratulate the team which has chalked out this program. I hope such programs will be continued in the campus, so that particularly our young researchers and the postgraduates and the PhDs will be benefited by this program. Wish you all the best. May God bless
you. Dr. Om Kumar, I have joined along with my colleague Dr. Nimmi. I just wanted to show before I correct my computer, I just wanted to show my face. I am Shastri. That's all. Very good morning to you, sir.
Thank you, Vice Chancellor, sir, for the inspiring words. Now, I would like to introduce the speaker of the day. Dr. R.V. Om Kumar, scientist, grade G, working currently as the head of the Molecular Neurobiology Group, Rajiv Gandhi Center for Biotechnology, Thiruvananthapuram, Kerala, acquired his MSc in Biochemistry from Kerala University, then his PhD in Biochemistry in the year 1992 from Indian Institute of Science, Bangalore. Later, his postdoctoral studies were done in Purdue University, USA, that is the initial 2 years, 1992 to 1994, and the remaining 2 years he worked in California Institute of Technology, USA. He has a research experience of 35 years, and his field of interest are in neurobiology, genomics, and biochemistry of calcium signaling. He has been awarded with many honors and awards. Just to mention a few, he received the Mrs. C.V.H. Rao Memorial Award for the best PhD thesis in 1992. He was the visiting scientist at the Health Technology Research Center, National Institute of Advanced Industrial Science and Technology, Japan in 2009. He was the UGC Visiting Fellow in 2012, SRTM University, Maharashtra. He was elected as Fellow of the Kerala Academy of Sciences. The patented invention of his research group was selected among the top 50 innovations for the year 2012 to 13 in the DST Lockheed Martin India Innovation Growth Program conducted by DST. He has 35 publications. He has given several invited lectures in India and abroad. He has 5 patents and has filed 3 more. He has guided 16 PhDs. With this brief introduction, I request Dr. Om Kumar to take over today's session on multi-target directed ligands for neurological disorders. I would also like to remind all the participants that at the end of the session, the feedback form will be posted in the chat box. You need to fill and submit the feedback form to get your attendance certificate. Over to you, sir.
Thank you, Dr. Nimi. Can you hear me?
Yes,
Audible,
sir.
right?
Yes, sir, you are audible.
Okay. Okay. Thank you, Dr. Nimi, for the nice words of introduction. Respected Honorable Vice Chancellor, Dr. Satish Kumar Bhandari, Dr. C.S. Shastri, the head of the department, and Dr. Nimi. I'm really honored to be visiting you virtually right now and talking about our research work to this august audience. I thank you for arranging this meeting and then calling me as a resource person here. I hope I will try to convey some points of relevance to you from the work that we are doing. I am not primarily a pharmacology person, but our research has taken us into to some extent of pharmacology also. So therefore, I'm sure I will, while I tell you about what we have, I should be able to get some feedback from you also. And then I look forward to more prolonged interaction with your great institute in the future, so that I can help you or I can get help from you. So I will share the screen now. Can you see the screen, Dr. Nimmi?
Yes, sir. Yes, sir. You have to
Okay.
put it in slideshow, sir.
Yeah, okay. Yeah, now it's in slideshow.
Yes, sir. Yes, sir.
Okay. I'm as introduced, I'm from this institute, Rajiv Gandhi Center for Biotechnology, which is in Thiruvananthapuram, very extreme south of the country. We are the capital of the state, Kerala. This institute started about 28 to near close to 30 years back. This institute started. And then I was one among the very first recruits to this institute. I completed 25 years in this institute this year. So I joined here with my training in the neuroscience, as well as before that as a biochemist. And then I started working on some problems related to the functioning of brain, particularly signaling in the brain, calcium signaling in the brain, in relation to learning and memory, as well as in relation to neurodegeneration. So what I will try to talk in today's presentation is, I'll just go into a brief introduction about neurological diseases with referring to Alzheimer's disease in particular, trying to highlight some of the general problems in addressing neurological diseases. And then I will go into the idea of multi-target directed ligands, which is one of the approaches taken in drug discovery. And then we'll try to tell you our experience and our opinions on that. And then I go into a very specific molecule called tacrin, and then how we have, through a collaborative approach, how we have tried to derive multiple compounds which are multi-target directed. And then we'll show you our data on the effect of these multiple derivatives of tacrin in vitro and in vivo studies, and how it right now is poised, and how it, what are its future prospects. Now, before talking about Alzheimer's disease, I just wanted to put a couple of words about our focal area, which is calcium signaling in the brain. And calcium signaling in the brain under normal conditions is very essential for synaptic learning and memory. There is an event called synaptic plasticity, which is very essential for learning and memory. And calcium signaling has been shown to play a major role in synaptic plasticity. However, there are conditions where the tight regulation of calcium signaling is lost. And once in many neurological conditions, the regulation of calcium signaling is lost, and then that will lead to excessive flow of calcium into the neuronal cells, and this is toxic to the cells. This kills the cells in a process called excitotoxicity, excessive excitation leading to toxicity. So our drug discovery approach has, our research interest primarily has been on calcium signaling, and therefore we have been trying to see how impaired calcium signaling in diseases can be targeted towards curing the diseases or towards managing the diseases. And for this purpose, we have developed some assay systems, and then we have also looked for compounds and molecules which can target impaired calcium signaling in these diseases, and thus see whether diseases can be managed. Okay, now coming specifically to the topic of today's talk, as I said, among neurological diseases, I have chosen Alzheimer's disease as one of the examples to tell you some general aspects of neurological diseases. Now, if you look at Alzheimer's, many diseases, neurological diseases, as well as other diseases, initially they may start with a single cause. Some may be multifactorial, but as the disease progresses, the disease will impair multiple systems in the body. So it may start in the brain, but slowly later it may affect the rest of the body also in multiple ways. So even inside the brain, it might start as some single cause, and we do not know what are we still do not know exactly what are the causes which will lead to a disease called Alzheimer's disease. Some are known, but not everything is known. But what we understand is that in Alzheimer's disease, once it is sort of full-blown, or once the disease is identified, generally the 2-3 major defects have been seen, and those are listed in this slide. Defect in cholinergic signaling, defect in glutamatergic signaling, and defect in amyloid protein processing. So you can see that there are multiple areas of defects in the disease, and therefore, if you want to manage the disease, you will have to correct the impairments in all these different areas or in these different mechanisms. Now, cholinergic signaling is basically neurons communicate to each other through neurotransmitter release and then reception of the neurotransmitter by an acceptor neuron, and that is how neurons communicate. Depending on the neurotransmitter molecule, different communicative signal systems are termed according to the neurotransmitter. Acetylcholine mediated signaling is called cholinergic signaling. So acetylcholine will be released from one neuron, and that would be received by this the successive neuron or the postsynaptic neuron through acetylcholine receptors, and that will lead to a signaling mechanism. And this signaling is essential in learning and memory. In Alzheimer's disease, this signaling is defective. Deficit in acetylcholine levels, the neurotransmitter level goes down, and that in turn causes cognitive decline, impairment in memory and other cognitive functions. Now, one of the approaches taken is inhibit an enzyme called inhibit enzymes which degrade acetylcholine as part of its normal cycling, so that acetylcholine, whatever is produced, would not get degraded, and thereby its level can be raised, and thus correct the defect. So the targets used are two enzymes, acetylcholinesterase, ACHE, and butyrylcholinesterase, BCHE. So if you inhibit these two enzymes, in fact, many drugs developed against acetylcholinesterase also block butyrylcholinesterase. And butyrylcholinesterase, it is a little broader in its specificity, but it does degrade acetylcholine. So by blocking these enzymes, acetylcholine degradation is stopped, thus acetylcholine level is increased, and thereby some amount of correction of the deficit in cholinergic signaling can be rectified. Some of the already used drugs in the clinics, which are cholinergic acetylcholinesterase inhibitors, are listed here: donepezil, rivastigmine, and galantamine. These are the major acetylcholinesterase inhibitor drugs which have been approved and are already being used in the clinics. Now, the other aspect that is defective is glutamatergic signaling. So here, the signaling is through the neurotransmitter glutamate. Glutamate released from the presynaptic neuron will bind to glutamate receptors on the postsynaptic neuron, leading to a signal, leading to neuronal communication. And this can also go impaired in Alzheimer's disease. The major mechanism by which this is impaired is because of increased levels of glutamate in the extracellular space. So as neurons degrade, as shown in this slide, here is an intact neuron, but here is a neuron which is degrading. As neurons degrade, a lot of their contents will be released into the cellular medium. And glutamate can be released into the cellular medium because of neurodegeneration. So this is not an impairment that is right in the very beginning of the disease, as we think right now. So this is a mechanism that starts operating a little as the disease progresses a bit. So neurons start degenerating, glutamate level increases. Now this excessive glutamate will cause excessive glutamate signaling. So intact neurons will keep receiving a lot of glutamate on their receptors. So excessive glutamate signaling would happen, and glutamate signaling involves calcium influx into the cell. Some of the glutamate receptors are calcium channels. Here is one receptor mentioned, Nmda receptor. This is a glutamate receptor which, upon binding glutamate, will open a calcium channel. And when glutamate level is high, it will keep on activating the calcium channel continuously in an unregulated fashion. So calcium flow into the cell also would become unregulated. That leads to the calcium toxicity that I mentioned earlier, excitotoxicity. And thus, neurodegeneration would happen, neurons will die. People have tried to target this defect by using blockers of Nmda receptor. If you block Nmda receptor by an inhibitor, then the glutamate signaling can be blocked. So even if excess glutamate is present, the glutamate induced calcium influx into the cell can be prevented to some extent. And thereby some amount of relief is possible, some amount of reduction in neurodegeneration and some amount of progression of the disease, the retardation on the progression of the disease can be achieved. One drug that is being used in the clinics which blocks Nmda receptor is memantine. It's a clinically approved drug. But as you can see, there are very few drugs which are currently available which can be used for Alzheimer's disease in the clinics. Generally, people give a combination of a cholinergic inhibitor and the glutamatergic inhibitor memantine. This is one of the common way in which Alzheimer's patients are treated, so that they get some improvement in their symptoms, they get some prolonged, their life gets prolonged, or the morbidity can be reduced to some level. Now, currently also drugs are in trial, but no drug is available, which target a third process which is impaired in Alzheimer's disease, that is amyloid protein processing. The protein processing goes wrong because of an enzyme called beta secretase, which will process the amyloid protein in a wrong way, and a wrongly processed protein will lead to amyloid plaques, and that is what accumulates outside the neurons as plaques seen in Alzheimer's disease. So drug candidates are in trial targeting this enzyme called beta secretase, which is involved in the abnormal processing of amyloid protein. So as you can see, you will have to use different drugs to address different defects in this disease. But how about having one molecule which could target these multiple pathways? This is the idea of multi-target directed ligands. Can we develop ligands which can we develop molecules which can block these different targets, and so that we can manage the disease by providing one drug. Now, what is the advantage in doing that? We already have multiple drugs, so you can give these multiple drugs and then do the same thing. What is the advantage of having a single molecule which can target multiple proteins inside the cell? So a few of these advantages are listed here. Lower probabilities of drug-drug interactions are which are encountered normally in combination therapies. When you do these combination therapies, one has to see that the drugs do not have contraindication. Drug-drug interaction, there should not be deleterious or harmful drug-drug interactions. If you have a single molecule, then that question doesn't arise. You're only putting one molecule. And off-target interactions are generally found to be less in multi-target directed ligand therapy. However, the one challenge would be the drug designing would be more complex. We are thinking of one molecule which should have pharmacophores which can go and bind to different proteins. So you need to design compounds in which multiple pharmacophores are present. At the same time, it should be very specific to only those targets of interest, and it should not go have off-target effects. So drug designing would be more complex in this case. Done with bioinformatics, which is very well developed in these days. So one can design multiple molecules in silico and then do a lot of iteration on their properties, improve the designs, and then narrow down to a smaller set of molecules, and then start making those molecules chemically and then testing them. So it is now these days possible to have a good amount of drug designing. So therefore, it is a worthwhile attempt to go after multi-target directed drug design. Now, another most important advantage with multi-target directed ligands is that if you have for each drug, for each molecule, before it becomes a drug, one has to study its safety profile, and that is a major part of a drug development program, as you all should be aware. We get lots and lots of inhibitor molecules which show activity in the in vitro systems, and then maybe even in the animal model, but then very, very few of them finally reach the clinic because they might show some side reaction or some adverse effects in the animal model, or sometimes when it is tried in the humans. So safety profile of a drug is extremely important before a drug reaches the clinics. So for each molecule, to get one molecule, you'll have to start with several molecules, and then you have to go through all the safety profiles, which takes several years, 10 to 15 years before a drug finally reaches the clinic. Now, if you think about 2 or 3 drugs towards one disease, say in Alzheimer's disease, cholinergic inhibition, glutamatergic inhibition. So different, if you are targeting only cholinergic inhibition, you have to develop one drug there, another drug for glutamatergic signaling. So we are talking about this long process of drug development being multiplied when you have individual drugs. But if you can think of one molecule having targeting on both these signaling, both these internal targets, then the safety efficacy, safety studies would be for one molecule, and that reduces the total amount of efforts involved and the total amount of cost involved in a significant way. The cost involved in as well as the time involved in drug development is a major challenge in drug discovery. And if that can be reduced by twofold or threefold, depending on how many targets you are combining, so that will be a major advantage in multi-target directed ligands. So having these thoughts in mind, we set out towards some multi-target directed ligand designing. So we chose this molecule called Tacrin. The structure is shown here. This is the molecule. The molecule that we have used is shown here. But this is the first approved drug for Alzheimer's disease. The first drug that was approved, and then people started using clinically for Alzheimer's disease. And this was initially an inhibitor of acetylcholinesterase, which is shown here. It is an inhibitor of acetylcholinesterase. However, although it showed some efficacy, it also showed hepatotoxicity, liver toxicity. And because of that, the drug was immediately withdrawn from the clinics. So it did not succeed. Although it succeeded in becoming a drug, it did not succeed in staying in the clinic. It had to be withdrawn because of hepatotoxicity. So it's primarily an inhibitor of acetylcholinesterase, one of the pathways defective in Alzheimer's disease. It also has weak antagonism against Nmda receptor, the other pathway which is impaired, glutamatergic signaling. So our aim was to see, can we improve a Tacrin molecule? It has come out, so it has succeeded in becoming a drug, but then there is some defect, that is hepatotoxic property of this, which escaped the drug discovery process, which escaped the scrutiny, and thereby it had to be removed. So can we rectify that? So how can we think about, how can we rectify that? So we thought, so there were two things that we had in mind. One is it possible to reduce the hepatotoxicity? Number two, it has weak antagonism towards NMDR, in addition to its strong action on acetylcholinesterase. Can we improve its NMDR antagonistic activity? So if we can make it a better inhibitor of NMDA receptor without affecting its acetylcholinesterase activity, then we are getting an efficient multi-target directed ligand. And additionally, is there something that we can do to reduce its hepatotoxicity? So for the toxicity, toxicity is always a function of dose. So any molecule can become toxic with high dose. Whatever is considered as safe is basically, safety is also a function of dose. Anything that is very safe in excessive quantity can become toxic. So in other words, whatever is toxic right now, if you can reduce the dose significantly, maybe its toxicity can be reduced, can be brought below the threshold, safety threshold. So is it possible to reduce the toxicity by reducing the dose? If you have to reduce the dose, then you will have to increase the efficacy. A dose is given because the drug is active or efficacious at that particular dose. So if we can design modifications in the drug so that it becomes much more efficacious, then it may be effective at a lower dose. And thereby you can reduce the dose, and that dose reduction, can it reduce the toxicity and then bring it into within the safety threshold. So this was our approach. So enhance efficacy for dose reduction and thereby reduce the toxicity, and also try to design efficacy enhancement. In this case, we have thought of enhancing the efficacy for Nmda receptor, along with if acetylcholinesterase efficacy increases, it is good. It is already a good target for acetylcholinesterase. So if we can also improve its Nmda antagonism or inhibitory activity to be enhanced, then maybe we are having an effective multi-target directed ligand, and with its increased efficacy, perhaps we should be able to get rid of its toxicity. So we had collaboration from a bioinformatics group and a chemical synthesis group. So what we thought is we will look at this molecule, derivatize, design derivatives, and then make those derivatives, and then test them in biological systems, how well they perform in the actual biological system. So by derivatizing, we were planning to improve its target interaction so that its affinity improves for the targets, particularly for Nmda receptor, which is a weak, which is an important pathway, but the molecule was weak against NMDR. At the same time, its ADME properties will have to be maintained, because this is a molecule which has already been tested for its ADME properties. So the nucleus has succeeded in favorable ADME properties. So by making these modifications, we have to make sure that the original ADME properties are not severely impaired. And also it's blood, for any neurological drugs, you also have another thing, another criteria to meet, that is its blood-brain barrier permeability. Not everything will be allowed inside the brain. So the molecule should be blood-brain barrier permeable. So tacrine is already blood-brain barrier permeable. The derivatizations should not affect it. So this were our challenges and then our approach. And we went ahead with designing the molecule. So our bioinformatics colleagues from the University of Kannur and Riken, Japan. So two groups were there doing this designing. So they did what is called as ensemble docking of this original molecule tacrine into candidate structures of Nmda receptor and acetylcholinesterase. So, so this ensemble docking involves considering the many different structures of these proteins. A protein is very dynamic. We may get a crystal structure of a protein, but in solution, a protein will be very dynamic. The crystal structure will keep changing, and then it will undergo minor alterations. So these alterations in structure in solution can be simulated by molecular dynamics simulations, and the many different altered structures of the molecules can be fitted with the drug, and then to find out which is the best fit structure and which is the best fit condition for the compound. And for all these different docking conditions or docked complexes, the binding energies would be calculated, and then some of them would be selected out from that with the good binding energies, and then they would be further evaluated. So about 75 derivatives were designed in our activity. Our bioinformatics collaborators designed 75 inhibitors, and they generated bound complexes on these 75 inhibitors, and then they looked at the binding energy. Ensemble docking was performed on the designed ligands, and binding energies were calculated, and we arrived at on the right side the actual data is shown. So I do not want to go. So the many different criteria in the drug designing, bioinformatics based drug designing are shown here. So looking at through all these different criteria, they have finally arrived at 75. They had 75 designs, and they had finally arrived at a smaller number of molecules, which looked very favorable and very very promising towards synthesis and then testing. So finally, a few of them, their ADME properties were also predicted by in silico methodologies. These days, there are in silico prediction tools to even predict ADME properties, which to a good extent will help us remove some of the very definite candidates which may not pass the screens. So we can remove major prima facie or obviously harmful drug designs in that screen, and then narrow down to a smaller number which have a chance to pass through the ADME screens. So finally, a few of them were selected. About 20-25 of them were selected, and they were synthesized. Now, in their synthesis, what they found is that in the molecule, tacrine molecule, they found the possibility of derivatizing at two positions. This position R1 position and R4 position, and with two positions to derivatize, they classified the compounds which can be made into three. So those which are derivatized only at R1 position, those which are derivatized only at R4, and those which have derivatizations in both. So group 1, 2, and 3. So 3 groups of molecules were designed. And then among them, some were synthesized by the chemistry group, and then they were tested in the biological systems. Now, coming to the biological systems that are to be tested, as I mentioned, we had 2 biological systems in our in our agenda, and that is, one is the acetylcholinesterase enzyme, the other one is the Nmda receptor molecule, which is a membrane channel, which is a neurotransmitter receptor. Acetylcholinesterase is an enzyme. There are standard assay methods to measure acetylcholinesterase activity. So using that assay, all these compounds can be tested for their ability to inhibit acetylcholinesterase. Now, Nmda receptor is a little more difficult because Nmda receptor is a calcium channel. And calcium channels are not, these are membrane-bound proteins, and their activity is calcium conductance. So they bring up several challenges in terms of their activity measurement. And drug discovery involves activity measurement be done several times if we have to screen multiple drug candidates. In such a case, one needs an assay method which is rather simpler and easy to perform, so that large number of compounds can be tested. So, Dr. Nitte, is everything okay? Are you hearing and seeing?
Yes, sir. Yes, sir. You are audible and everything is fine, sir.
Okay, fine.
You can continue, sir.
Yeah. Okay. So, so Nmda receptor is a calcium channel, and calcium channel comprises a significant portion of ion channel targets. And the general approach to drug discovery for calcium channel is either structure-based design, that is, you look at the structure of the protein and then design the molecule, the one which we have done right now. One can also do high throughput screening of libraries without much design, make large compound libraries and then screen them. To do this kind of a screening, particularly, even when you design, one ends up with several molecules which are probable, but the number of molecules can be lesser in a design-based approach. However, in a blind screening approach, the number of molecules to be tested would be much larger. So in either way, the method of testing the molecule on the biological system is going to be used multiple times, or you need what are called improved throughput, high throughput assay systems to do test these molecules. And for a high throughput screening, the assay being used would be it would be good to have the assay system which is that can be carried out easily, and it would be preferred if the assay system is cost effective, if it is not very expensive. Now, how are calcium channel assays done in general? Calcium channel assays, Nmda receptor being a calcium channel, I'm just saying calcium channel as a general, in a general perspective. Nmda receptor is a calcium channel, and calcium channel assays are generally done either by electrophysiology or by real-time calcium imaging. So electrophysiology is a technique with which current across membranes is measured with using very sensitive electrodes. So techniques such as patch clamp, etc., are used for current measurements across membranes. Calcium channel allows calcium ions to flow through the membrane, so it's basically a calcium current that can be measured with an electrode. So this is electrophysiology. So a calcium channel activity would generate a calcium current, and you measure it as the activity of the channel. The other method is real-time calcium imaging. Calcium which is coming inside the cell can be detected by using fluorescent probes, which will bind calcium and then give fluorescent signal. So you don't look at the channel, but you look at the calcium that is coming inside the cell in response to activation of the channel with its specific agonist or ligand. And then use imaging techniques or fluorimetry techniques to capture the fluorescent signal in response to calcium by using fluorophores or fluorescent probes. Both these techniques are what are called real-time techniques. In other words, in these techniques, the activity has to be observed as and when the activity is happening. When the enzyme is working itself, one has to observe the enzyme and then capture the signal for the activity. So these are called real-time techniques, as opposed to endpoint techniques where you can do an enzyme activity assay and then stop the reaction, finish the reaction and stop the reaction, then measure how much product is formed over a course of time. So that is a class of assays called the endpoint assays. There you don't need to observe them while the enzyme is working. You can allow the enzyme to work for some time, then see how much product is formed. And then since we know how much time it has worked, so you can calculate the amount of product formed per unit time and thereby calculate the activity. So endpoint assays are usually easy to perform. Real-time assays, where you have to capture the activity as and when it has happened, they have to be much more fast and much more sensitive. For example, in this case, calcium imaging, the calcium that comes inside the cell will give a fluorescence, but this fluorescent signal is very short-lived because cell cannot live with high levels of calcium for a long time. The calcium that is coming into the cell will be immediately absorbed or taken up by intracellular or calcium stores, so that the amount of free calcium is immediately reduced. And that is how cells manage calcium toxicity, or they will prevent calcium toxicity even when they use calcium signaling as a major signaling mechanism. So therefore, calcium signal induced fluorescent signal also will be very short-lived. So if you activate a calcium channel, a calcium flux would have go into the cell. If you have used a fluorescent probe inside the cell, then it would give a fluorescence, a flash of fluorescence when the calcium is high, and the calcium will be immediately absorbed, so the fluorescent signal will be gone. So the equipment that you are using, the microscope or the fluorimeter, has to be a very fast, has to be very fast in its data capture. It has to be very sensitive because the fluorescent signal sometimes can be very mild. So the equipment has to be very sensitive. Particularly electrophysiology is where the currents through the channels are measured. So the electrodes have to be extremely sensitive. So real-time techniques, therefore, are difficult to perform compared to endpoint assays. In an endpoint assay, many of the colorimetric assays that we do in the labs, they are all endpoint assays. They are much more simpler. So, and that is why you can see in any educational program or in any practicals, students are first taught endpoint assays. They start with endpoint assays because that is manageable and that can be shown in a lab very easily. The real-time assays are much more advanced assays or advanced methodologies. But but real-time assays have certain major advantages, is that they have high temporal resolution and high spatial resolution. In other words, they are very fast, which I have already talked. Even very fast events can be captured, and they have high spatial resolution. In a cell, if calcium release happens in some subcellular region, say from mitochondria, some calcium is released, or from the endoplasmic reticulum in one region, in the cell body of the neuron or the axon of the neuron, there is calcium release, the imaging technique can pick it up. So spatial resolution will also be high. However, the disadvantages of real-time assays are that this real-time data acquisition necessitates the need for very expensive instrumentation. The equipments will have to be very fast and very sensitive, and obviously then they would become very expensive. And because of these expensive equipments, and also because of the technical technical challenges in doing this, in performing this assay method, high throughput scaling would also be difficult. It will not be possible to repeat this assays multiple times that easily compared to an endpoint assay. So, and calcium channel assays are done only with real-time assays. Until we first came up with a method which is not a real-time method. So our this is about maybe 10 years back when we came up with this new assay for calcium sensing. So the reason for that is we have been working on calcium signaling with relevance to the role of calcium signaling in neuronal function and neuronal function in normal condition as well as in disease conditions. So we had some systems developed, and then we found that they can be used for calcium sensing. And interestingly, what we found is that it is possible to perform an endpoint assay for calcium sensing using a protein-protein interaction between two proteins, calcium calmodulin dependent protein kinase 2 or CAMK2 and Nmda receptor, a subunit of Nmda receptor called NR2B, which I will be calling gluN2B later, because this is the earlier terminology. Now it is called gluN2B. So interaction between these two proteins can be used to detect calcium, and the calcium infects into the cell in an endpoint assay format. So without a real-time assay. And this was the first endpoint assay for calcium channel that we developed in our lab. So the principle of the assay is based on this cellular event that happens in the post-synaptic compartment, in the synapse, that is in the accepting synapse. So it has these neurotransmitter receptors. What we've shown here is Nmda receptor. It will bind the glutamate, which is released from the presynaptic neuron, and it will open a calcium channel. Calcium will flow into the cell. It will bind to this protein called calmodulin. I'm sure you are all aware. And calmodulin calcium complex can now go and activate many proteins. And one of them is this protein kinase called calcium calmodulin activated protein kinase type 2 or CAMK2. This is a major protein highly enriched in the neurons and particularly in the synapses. When activated, this protein, which is normally in an inhibited condition, would get its auto-inhibitory domain, which normally blocks its active site, removed from the active site because of binding of calcium calmodulin complex, and thus the active site becomes exposed. Now the protein can act on its substrates. So this is the mechanism of activation of this protein. Now, along with this activation, a second site on the protein also gets exposed or opened up, which was otherwise blocked in the inactive enzyme. And this is a binding site. And by using this binding site, this protein can bind to Nmda receptor. This subunit of Nmda receptor called gluN2B. And because of this binding, the activated kinase would now move towards the membrane and then will bind to Nmda receptor. Now, the interesting aspect of this binding is that this is an extremely strong binding, that it is almost irreversible. It doesn't fall back even when the calcium calmodulin complex goes away from the protein. The calcium, as I mentioned earlier, calcium level would be high only for a short time. It will immediately be absorbed by intracellular organelles. And when the calcium concentration goes down, then calmodulin bound calcium will also fall off. It will be released. So without calmodulin, without calcium, calmodulin will not be active. It will fall off the enzyme. So when this complex falls off the enzyme, it should normally go back to its inactive state. But once it binds to, during this activated state, if it can bind to Nmda receptor, then even if the calcium calmodulin complex falls off, the enzyme will not go back to its inactive state, or enzyme will not dissociate from the bound state. So it would stay permanent in this complexed form. So this is an important and a very interesting or a useful property of this binding, which we could make use of. So if we see inside the cell the presence of this complex, that says that there was a calcium influx. normally the enzyme by itself would not go and bind. If the enzyme has gone and bound, and if you detect this bound form, that reports that there was a previous calcium influx. So if during the activation, if you put an inhibitor, and supposing the activation did not happen, calcium influx would not happen, so this complex would not be seen. So by observing this complex, we can say whether calcium influx happened, and thereby we can say whether the calcium channel was active or not active. So in presence of an inhibitor, whether a calcium, in presence of a compound, whether the compound is inhibiting the channel or not can be can be inferred by seeing the presence of this complex. Now the assay this was the principle of this assay that we used. So what we have slightly adapted the system. We have taken this enzyme CAM kinase, CAMK2, and this Nmda receptor subunit, and then put them into a non-neuronal system, a heterologous system. The system that we used is HEK293 cells, human embryonic kidney cells. So this is a non-neuronal system. The aim of using this system is that it's easy to culture. Neuronal cultures are more difficult, whereas HEK cell is a cell line, so it is easy to culture. Secondly, it doesn't have background expression of these two proteins, CAM kinase and Nmda receptor. So CAM kinase was fused with GFP. So there was some engineering done, some protein engineering done. So CAM kinase was fused with GFP, and Nmda receptor, the whole subunit, we have cut out only the fragment that is needed for binding, and then fused with a mitochondrial targeting sequence. So both were done with the genetic engineering, and the mitochondrial targeting sequence would make sure that this goes and binds to mitochondria if you express it inside the cell. So the cDNA for the GFP CAMK2 and the Nmda receptor fragment with mitochondrial targeting sequence were expressed in the HEK293 cells by transfection with the plasmid vectors carrying these constructs. And a cell carrying both these proteins, if it receives a calcium influx, as shown here, it will go and activate CAM kinase. The inactive CAM kinase will now get activated, and the active CAM kinase would now try to bind to Nmda receptor. And as you know, the Nmda receptor has been designed in such a way that Nmda receptor fragment has been designed in such a way that it will now express on the mitochondria. So CAM kinase will go towards mitochondria and then bind to Nmda receptor. So in response to calcium influx, what you would see is that the CAM kinase, which was present in the cytosol, would now accumulate around the mitochondria. And this signal would be stable. It will not fall off when the calcium channel, when the calcium signal goes away. And you will be able to observe the signal for calcium as accumulation of this protein, which is labeled with GFP. So accumulation of GFP fluorescence around mitochondria. And this is how the assay will look. So in the top, on the top left panel, what you see is cells which have been transfected with both these proteins, the GFP CAM kinase and the Nmda receptor fragment that is mitochondrially targeted. GFP CAM kinase is distributed in the cytosol, so you can see fluorescence of green fluorescent protein throughout the cytosol. If you activate the Nmda receptor, which is also expressed on this cell line by transfection using its ligands, glutamate and glycine, in presence of extracellular calcium, then you see that the fluorescence which was present on the cytosol accumulates onto the membranes, onto the intracellular organelles. And this happens only if you have glutamate, glycine, and calcium. Glutamate, glycine to activate the receptor, and calcium, of course, has to be present on the extracellular medium so that it can flow in. If you add glutamate and glycine without calcium, minus calcium, it will not happen. Now, this activity can be blocked if you do this activation in presence of known inhibitors of Nmda receptor. And that is what is shown here. Glutamate, glycine, and calcium would activate the receptor. So calcium goes in and then causes accumulation of fluorescence around the cells, on the intracellular organelles in the cells. Now, if you do the same activation procedure in presence of an inhibitor of NMDA receptor called AP5, then that does not happen. So the accumulation of fluorescence does not happen. The same thing happens if you use another inhibitor of NMDA receptor called MK801. There also the activation procedure that is normally seen by activation of NMDA receptor would be blocked. So this is how you can identify an inhibitor. You do the activity in presence of an inhibitor, then you will not get this this localization pattern, or we call this the punctate appearance. You will not see this punctate appearance. So if a compound is not an inhibitor, then you will see this punctate appearance. So absence of this punctate appearance in an activation indicates, yes, there is an inhibitor. So this assay system was developed in our lab, not as part of this project, but we had developed it already and we had it. So we used this assay to test the compounds that we made, the tacrine derivatives that were made, to see whether they can inhibit NMDA receptor. And then a series of screening assays were done, both for NMDA receptor as well as for acetylcholinesterase. And what we found is that, so these are the numbers shown here are the derivatives that were shortlisted and then actually synthesized. And these derivatives that were tested along with the precursor molecule, which is tacrine, which is shown here. So tacrine's inhibition is, or IC50 value for inhibiting acetylcholinesterase was around 100, and the various other derivatives showed varying degrees of inhibition as seen by the IC50 value, the 50% concentration, concentration required to inhibit 50% of the activity. The higher the IC50, the potency is less. The lesser the IC50, that means the inhibitor is a very potent inhibitor. We can see that there are some derivatives which had improved potency compared to tacrine, the original molecule. So there are a few compounds which had improved inhibitory potency. The compounds were tested on butyrylcholinesterase also, and we found that many of them were inhibitory on butyrylcholinesterase also. They caused, what is shown here is the actual IC50 value. They were all inhibitory on butyrylcholinesterase also, but primarily it is the acetylcholinesterase inhibition that was of interest to us. Our aim was mainly to make sure that these derivatizations would not take away the inhibitory activity. That would maintain the acetylcholinesterase inhibitory activity, but interestingly, we found some have even improved in the acetylcholinesterase inhibition. Now, these derivatives were then put on the NMDA receptor assay. And if you remember, tacrine, the original compound, was a poor inhibitor of NMDA receptor. And you can see that original compound tacrine shows inhibits NMDA receptor only to a small extent. This is percentage inhibition on the y-axis. But among the compounds that were made, there are a few which did not improve, which are shown here. Their inhibition was only as good as tacrine, which means they did not really attain NMDA receptor inhibitory activity, but there were many which became very inhibitory on NMDA receptor. Their percentage, they were inhibiting NMDA receptor to a significant extent. And this is, here is a representative image of the actual assay. Normal NMDA receptor activity would give you this punctate pattern. And in the absence of calcium, you would not see that punctate pattern, which you are seeing here. I hope you can see this images clearly. Dr. Nimi?
Yes, sir. Yes, sir. It's clear.
Yeah. So this punctate pattern is blocked. Without calcium, it will not happen. And with all the conditions, if you include an inhibiting compound like 211 or 208, you can see that the punctate pattern is blocked. It is not forming. So 211 and 208 are two compounds which showed good inhibition of NMDA receptor here, 211 and 208. They caused 100% inhibition of the activity at even 20 micromolar concentration. 20, 50, and 100 micromolar were initially tested, and they were causing 100% inhibition even at 20 micromolar concentration. So they were very potent inhibitors. So similarly, all these others were tested in this kind of an assay, and their inhibition was identified. Now, we have a way of quantifying the activity in this assay. You can count the number of cells, and then the reduction in the number of punctate cells was correlating with the extent of inhibition. And this was already developed. So we used the quantification method using this assay, and then all these compounds were subjected to Ic50 determination. That is, the compounds were given at multiple concentrations. Each compound was given at multiple concentrations, and the activity profile was taken, and from there Ic50 value was calculated. And you can see that for these selected compounds, we had selected 14 compounds finally. Their Ic50 values are indicated here. Compared to TAC, all their Ic50 values were in the range of about maximum that we found was 40 micromolar, near 40 micromolar. That means the lowest potency. The weakest inhibitor had an Ic50 of 40 micromolar, whereas the strongest inhibitors were extremely low. The Ic50 values were in the range of about 0.3, 0.2 micromolar, or 300 to 200 to 300 nanomolar concentration. So there were extremely potent inhibitors also among the compounds that we made. And when you compare this to the Ic50 of tacrin, tacrin had an Ic50 which is reported in literature is about 250 to 300 micromolar. So you can see that all these compounds were much better compared to tacrin in their inhibitory ability. The weakest ones were still about 5 to 6 fold better than tacrin. 250 versus 40. 50 was about 5 to 6 fold better than tacrin, and the ones which are very strong, 0.3, 0.4, they are about 500 fold better than the original compound tacrin. So we were fortunate in getting some of these compounds to be very potent in blocking Nmda receptor activity, whereas the original molecule tacrin was not as good as a Nmda inhibitor. Now we just tested so we have been able to engineer Nmda receptor activity onto the tacrin molecule now by the derivatization. And these molecules did not lose their original activity, which is acetylcholinesterase activity. So they have now become bifunctional compounds, or they have become multi-target directed compounds. Rather, they have become effective multi-target directed compounds. The original compound had very weak targeting to Nmda receptor, which we have been able to improve. And we have now compounds which are effective against, strongly inhibitory against acetylcholinesterase as well as Nmda receptor. So having achieved this, now we were about to go into testing them in a more complex or higher biological systems. So before that, we just wanted to see whether we can check their hepatotoxicity. This was another problem that was there with tacrin, and we used an in vitro system for testing hepatotoxicity, for preliminary testing of hepatotoxicity. HepG2 cell line, a liver derived cell line, hepatocyte derived cell line, is generally used as a primary as a preliminary system for assessing hepatotoxicity. Any compound which is toxic to liver cells, then you can say that they would be toxic in the animal also. They would be hepatotoxic. They would cause liver toxicity. So we found that all these compounds, in comparison to the original compound tacrin, which was known to have hepatotoxicity. So with the 4 concentrations that were tested, you can see that tacrin causes some amount of cell death as the concentration is increased. Lowest concentration to higher concentration, there is the cell survival reduces to close to 50%. So tacrin shows its hepatotoxicity, whereas the compounds that we made, they all were less toxic. And even at the highest concentration, you see each of these is having 4 concentration. 4 bars are there, each bar showing 1 concentration. So even in the highest concentration, almost all the compounds were less toxic, or rather they were not causing any toxicity. They were all near 100%. So which means they did not appear to be hepatotoxic in this in vitro assay. So we then proceeded to test whether this Nmda receptor activity, inhibitory activity. So acetylcholinesterase activity is already proven for tacrin, and in vitro, in vivo studies have been done, and the literature is available. So we did not go more into acetylcholinesterase activity. We wanted to make sure that the modifications did not affect acetylcholinesterase activity. So we then studied in depth the Nmda receptor activity of these compounds. So we wanted to see whether Nmda receptor present in the neuronal cells, whether they would be inhibited by these compounds. Now, in this case, Nmda receptor was expressed in HEK293 cells, and then the activity was measured. So we went into a neuronal system, primary neuronal cultures, prepare neuronal cells from the brain of rat embryos, and then they can be maintained in culture for up to a month. And so we tested, did our assays after keeping the cells for about 9 days in culture. By that time, the cells would develop all the connections and neurites. Normally, when you prepare all these from the brain, all the cells, because of the preparation, there will be a lot of mechanical damage to the cells. So all the cells will be rounded. You will not see this neuronal morphology on the day of preparation. But as you keep them, they again regain their morphology. They sprout out their neurites and make connections, and then you get this neuronal morphology by about 9 days. So we used day 9 cultures for our assays. And what we have stained the neurons with a neuronal marker called a MAP2, microtubule associated protein. So MAP2 staining shows that we have neuronal cultures here. And if you treat this culture with glutamate, which is an Nmda receptor agonist, it's a glutamatergic signaling component. So if it will bind glutamate and then cause calcium influx. So a particular dose of glutamate and for a particular duration, the calcium influx would be excessive, so that it will cause cytotoxicity. And you can see that glutamate treatment is reducing the number of cells because of cell death. So this is the typical glutamate toxicity measured in vitro. Now, if you treat with glutamate in presence of a known Nmda receptor blocker, MK801, then you can see that this cell death is not happening because Nmda receptor is blocked. In the same way, we tested all the compounds that, some of the compounds that we developed, and you can see that in presence of most of these compounds, here we have tested only a few of them, and in presence of most of them, compared to the condition of glutamate-induced cell death, they are all showing protection of neurons. There is neurons are being protected from undergoing cell death. So of course, they may not match with MK801 protection, but they are indeed protecting compared to the excitotoxic cell death. So what we are showing here is that cell death induced by glutamate, which is known to be Nmda receptor mediated. We are also showing that standard Nmda receptor is blocking that. So these molecules are also blocking Nmda receptor mediated cell death or glutamate induced cell death, which means they are really functioning in terms of bringing out changes at the cellular level. So on the left side, several different assays, quantification methods were used to quantify this property, that is neuroprotection, allowing the neurons to survive in a glutamate toxic condition. So these compounds are showing neuroprotection under these different conditions. So here you can see, compared to control, glutamate treatment is causing a reduction in the number of cells, but in presence of the compounds, the number of cells is preserved. There is a good amount of reduction in the amount of cell death. So they are showing protection of neurons in an in vitro condition. So then we went, we took it forward to an in vivo model, the rat model, and we wanted to see whether this molecule, whether these molecules can protect glutamatergic toxicity in the, can protect animals or the brain in the in vivo condition against glutamatergic toxicity. There is excessive glutamate-induced neuronal death, which is one of the components in Alzheimer's disease. So we used the model in which rats were injected with this compound, monosodium glutamate. So it's basically glutamate, a salt of glutamate. So by injecting glutamate, we are trying to increase the level of glutamate in the brain. Normally, this happens because of various other reasons in the brain in the disease condition. Because of degenerating neurons, a lot of glutamates will be released. Then glutamate uptake mechanisms will be defective. Therefore, the glutamate in the extracellular space will maintain. So there are reasons because of which glutamate level will be high in a disease condition. So we try to generate high extracellular glutamate by injecting monosodium glutamate. And the injection was done along with the injection was done to the animals which were also treated with the inhibitors. And we tried to assess these animals then how the symptoms of neurodegeneration or brain damage in those animals show up in presence of monosodium glutamate, and then whether that could be prevented by using these compounds that we developed, which are Nmda receptor blockers. So animals are injected for 15 days with the monosodium glutamate, and the first assay was their behavior was studied from day 10 to 15. So there are several assays, the behavioral assays for animals, which reflect their brain function. And behavioral assays can be used to detect damage to the brain or pathology in the brain. Brain damage will usually lead to abnormal behavior, and that is what we used. So the test, the behavioral assay that we used is called the Morris-Watermaze test. For those of you who are not familiar, this is a test where a rat is put into a tank of water, which is the circle shown here. It's a tank, a water tank filled with water, filled maybe 75% with water, and then it will have a hidden platform which will be just below the water level. And then if you drop a rat into the water here, the rat will swim around the tank and then will try to come out of water. So initially it will try swim through the margins of the tank and then try to jump out, but it won't be able to do that. And then it will start exploring, and then finally it will find the platform in the inside the tank, which is not visible, but a little bit underwater. But the rat, by trial and error, by moving around, it would find that yes, there is a platform there. And then once it finds the platform, it can stand on the platform. So that is very convenient for the rat. So it would prefer to stop swimming and then stand on the platform. Now on day one, when you drop the rat, it does not know whether a platform is there and then where it is. So it will keep swimming around for a long time before it can find the platform. Now you repeat this trial. Once it finds the platform, you remove the rat, and then again you put it back into the water. So thereby you keep giving it multiple trials, and through these multiple trials of practice, the rats slowly learn that there is a platform and then where the platform. So after repeated trials for 5 days, by day 3 onwards, they start learning that there is a platform. And on day 5, you can see that the same rat which was moving around like this on day 1, on day 5, the moment you drop it, it will straight away go to the platform. So this shows that the rat learned that there is a platform, and then the location of the platform is here, so that where you drop it in the tank, it will straight away go to the platform. It knows, so it has learned, and this is going through 5 days. So on day 3 onwards, they start understanding about the platform, and then the subsequent days, if you bring it and then drop it, it will the first trial itself on a new day, it will straight away go to the platform. So that means it is remembering. So the time taken by the rat to reach the platform is taken as a parameter called escape latency, and then the escape latency is correlated to its ability to learn and memorize. Now, animals which are injected with saline learn well by day 5, but animals which are injected with monosodium glutamate, the compound that we talked now, that means the animals which are overloaded with glutamate continuously, On the 5th day, they still do not perform well. They still take more time to find the platform. Compared to the untreated animal, they are taking much more time to reach the platform. That means there is impairment in their brain function. Their ability to learn and memorize is impaired. Now, if you do this treatment in presence of a known Nmda receptor inhibitor, which are already known to be effective in vitro and in vivo, you can see that we used a compound called dextromethorphan. It's a drug which is being used as an anti-inflammatory or against allergies. It is an Nmda receptor inhibitor. If you treat the animals with dextromethorphan along with monosodium glutamate, you can see that compared to the control, the monosodium glutamate treated animal learns equally well because they are also treated with dextromethorphan. So the monosodium glutamate induced hyperactivation of Nmda receptor does not happen here because along with monosodium glutamate, dextromethorphan also is going to the system. So increased activation of Nmda receptor is blocked by dextromethorphan. So if this excessive activation of Nmda receptor is blocked, then the corresponding brain damage would also be prevented, and therefore the animals are not affected. So their learning ability, their performance in this Morris-Watermaid test is also unaffected. So these animals, if you look at their brains, so basically I'm showing data on the animal model, basically characterizing the animal model to see that this animal model is suitable for testing Nmda receptor inhibitors. So in this animal model, if you take the brain of the animal and then look for some of the cell death associated molecules, TrkB is a cell death associated molecule in the neurons. When there is toxicity, TrkB levels will go down. You can see compared to the saline treated animal, the monosodium glutamate treated animal, there is a reduction in the level of TrkB. And that is quantified and then shown here. Multiple experiment from multiple experiments, if you quantify, there is a reduction in TrkB. And this reduction in TrkB induced by monosodium glutamate can be prevented if you also treat the animal with dextromethorphan. Please ignore the middle panel. Let's look at the right extreme panel. Along with monosodium glutamate, if you also feed dextromethorphan, that is a Nmda receptor blocker, the TrkB reduction is prevented. So there is no major reduction in TrkB. So just like what we found in the behavioral assay, where the monosodium glutamate induced impairment was prevented, in a biochemical parameter measurement also, if you looking at the brain, inside the brain also, we find that a change which is induced by monosodium glutamate, which is shown here, is prevented by dextromethorphan. So monosodium glutamate induced change is therefore mediated through Nmda receptor, and an Nmda receptor blocker is able to prevent it. So this animal's model therefore can be used to test for other Nmda receptor blockers. So we used the compounds that we developed and then administered them to the animals along with monosodium glutamate. And we measured their behavior. And this is the behavioral analysis data. So on the left extreme panel, the test on a few of these compounds. Not all the compounds could be tested so far. We have tested only a few of them. Okay, let's look at this figure first. On this, the lowermost right lower panel, panel D, you can see that the saline treated animal on day 1, it is moving around for a long time, and here is the platform. On day 3, it reaches the platform from the starting point where it was dropped. It reaches the platform with less amount of exploration. So it has learned to some extent. On day 5, it has learned pretty well. The moment it is dropped, it reaches the platform straight away. So it has learned well. The monosodium glutamate treated animal, on day 1, it is like any other animal, but on day 3, there is no significant improvement. Compared to the saline treated animal, it is compared to that, it is taking much more time. It is moving around for a long time before it reaches. So this is showing the track taken by the animal. the animals, while they are performing, they are dropped into the tank, they are also under a video camera. So the video recording can then be processed so that the track taken by the animals can be extracted out. That is what is shown here. And on day 5, these monosodium glutamate treated animals are still impaired. They are showing some improvement, but they are far less efficient in finding the platform compared to the saline treated animals. So their learning memory function is impaired, or their brain is damaged. Now, if the monosodium glutamate treatment was given along with one of the compounds, the compound name given here is 5A, but it is actually, the same is shown here as 201. Both are the same. So it's the same compound. So this compound, if you administer along with monosodium glutamate, you can see on day 3, it is better than monosodium glutamate alone. And on day 5, of course, it is better than monosodium glutamate alone. So monosodium glutamate impairment is prevented if this compound is also given along with monosodium glutamate. And it is coming closer to the saline alone. It is not completely as good as the saline, but it is much better than the monosodium glutamate treatment. And this is the compound alone. If you administer the compound by itself, it learns properly. So if you measure the total time taken by the animal on the 5th day to reach the platform, you can see saline takes less time, the MSG takes much more time, the compound treated one, compound with MSG, it takes lesser time. And that is shown in this plot here. In panel A, saline takes about 10 minutes on the 5th day to reach the platform. Monosodium glutamate treatment, it takes much more time, close to 30 seconds to reach the platform. It is the seconds, not minutes, 30 seconds to reach the platform. But along with monosodium glutamate, if you treat with the compound 201 is one of the compounds, it takes far less time. Here is another compound 208. Again, with the monosodium glutamate, it is taking far less time. So what is shown in between are controls where the compound alone is given. Two derivatives tested are showing significant improvement or protection of the animals from monosodium glutamate induced impairment. Now, if you use tacrin, the original molecule from which we made these derivatives, tacrin is not able to cause improvement. Monosodium glutamate treatment is causing the impairment along with tacrin. So, which means the tacrin, the original compound was not able to protect, but the derivatives that we designed are able to protect the animals against glutamate-induced toxicity. So, this was tested again in the same model with a higher dose of monosodium glutamate. There again, it is showing protection. So, we have therefore been able to show that the compounds that we designed are active against Nmda receptor. They showed Nmda receptor they prevented Nmda receptor induced cell death in primary neuronal cultures, and in animal models also, they are able to prevent Nmda receptor mediated neuronal damage. So, to summarize, what we have achieved is to generate a series of multi-target directed ligands. And I talked to you about the advantages of multi-target directed ligands in the first part. And we have adopted that approach. And then we used this molecule called tacrin, and then derived several multi-target directed ligands. And their efficacy was tested in these multiple systems, in vitro and in vivo. And we are now having a series of such compounds which are showing in vivo efficacy in an animal model. Now they can be taken forward in the drug discovery route. They can be tested in other animal models. More detailed studies on the animal models are being done now. Their toxicity is being analyzed. They can be, if they are non-toxic, then we can take them to further levels of drug discovery. So I want to acknowledge my group and my collaborators who have been part of this work. On the left is my research group, various research, various PhD students who have been part of this work, involved in the development of this assay system that in our lab. And then my collaborator, the Kannur University Department of Biotechnology, where Dr. Sadashivan is a bioinformatics person, and his student Ramya is the one who did most of these experiments. She came to my lab and then did most of these experiments after the chemical synthesis and bioinformatics were done in those respective labs. And this is the Japanese group which were doing the bioinformatics part. And this is the group in Andhra Pradesh, Guntur. This institute called VFSTR, Vignan Foundation for Science, Technology and Research. There in the chemistry department, Dr. Sheikh Anwar and his student Koti Reddy were involved in synthesizing these compounds. I also want to acknowledge the funding from my institute as well as from DBT. And also I had technical support from some staff in our institute. The names have got hidden here, Indu and Gigi. And thank you all for the patient hearing. Thank you.
Okay, thank you, sir, for the very educational and edifying talk on multi-target directed ligands for neurological disorders. Now the session is open for question and answer. Here we have got some questions, sir, from the chat box.
Yeah.
First of all, Mr. Omkar Mali has asked, first he asked a question, what are the in vitro models to check the activity of drugs against Alzheimer's disease? I think,
what are the
in vitro models?
Okay.
To check the activity of drugs against Alzheimer's disease.
Okay.
And then later he asked one more question. If we want to perform these assays for our drugs for Alzheimer's, how much will be the cost for these assays? And also he wants to know if any collaboration work is possible with you. Mr. Omkar Mali has asked.
All right. Yeah, so the as I mentioned, there are today I covered 3 major impairments in Alzheimer's disease: the cholinergic impairment, glutamatergic impairment, and amyloid protein processing impairment. So, so these 3 assays for these 3 enzymes can be used for in vitro screening for molecules for Alzheimer's disease. Acetylcholinesterase activity assay is one assay, activity assay of acetylcholinesterase. The second one is Nmda receptor assay. There are multiple ways by which Nmda receptor assay can be done. So I talked to you about the assay that we developed, but there are also other methods of assaying Nmda receptor. If you have just one compound to be tested or 2 compounds to be tested, you can use any of these assays. You can also go for the electrophysiology assays in a lab where that is done. But if you have several compounds, then it would become difficult to do it with those assays. There, the assay that we developed might be more handy. And then, of course, amyloid beta processing inhibition can be tested with beta secretase enzyme activity. That is again an in vitro assay. There are kits available for that. For the acetylcholinesterase activity also, there are kits available to do the assay. And collaborations, yes, they are possible, but usually collaborations are today's data that I presented is a collaboration in which the person from the lab came to our lab and then did the experiments. So that only could be possible. We have the systems in our lab standardized, so we will be able to provide the technical support as well as the systems are established. So anyone who is interested can think of coming here and then doing the experiments. So that can be discussed.
Sir, Ms. Harsha, a PhD student, wants to ask, based on what criteria derivatives will be designed and how to select the substitute groups to be added?
This is the bioinformatics part. they dock the compound, original compound, tacrin, into the protein molecule, and then they have selected a few docked complexes, and then in a few docked complexes, and then in the docked complex, then they look at what are the additional possible interactions from the tacrin molecule, from its two positions that I mentioned, R1 and R2. Are there some nearby amino acid side chains with which a hydrophobic interaction or an ionic interaction can be engineered? So you have to put some group onto the compound, which will now be reaching proximal to the amino acid side chain of the protein, so that a productive interaction can be engineered. So that is the approach they have done. So this can be done with only modeling and simulation. Basically, they have looked at, by putting the groups, they have put groups which can form hydrogen bonds. They have also put ionic groups so that some electrostatic interactions can be formed. And hydrophobic groups also have been tried, so that the additional hydrophobic interactions are engineered so that binding becomes stronger. Hope that answers.
Professor Om Kumar, I just, not a question, I was just wondering to whether I can discuss with you. See,
Yeah.
this is, we are trying to analyze the activity of acetylcholine level enhancement by inhibiting estrase enzyme or NMDA activity. Have you come across any procedures in which actually the brain imaging without invasion of the animal can be done? I am talking about non-invasive assessment of maybe the amyloid plaques or the tau proteins, or even any of these things in Alzheimer's, or the elevated level of acetylcholine in the brain by suppression of this one. A sort of imaging, we can image the brain and find out the concentration enhancement without actually. Right now, most of us, we are sacrificing the animal, taking the brain homogenate and trying to find out the acetylcholine estrase activity by actually measuring the acetylcholine level by Ellman method or whatever it is. I mean, the methods what you have described.
Right.
Now, I was just wondering, non-invasive methods, have you come across anywhere where actually you can put the rat or mice below some sort of scanner and find out the level of these neurotransmitters and other this one?
Yeah, no, there are in vivo animal imaging systems now available.
Yeah.
They can, in principle, use fluorimetric or luminescence based probes in the animal and then detect them. It is available. People have been using it to detect cancers. If there's a major cancer developing in some organ after an injection, they are able to detect that. But for the brain, whether a change in the level of a neurotransmitter, I do not know whether such techniques are available, but if there are suitable probes available, maybe we have to check the literature whether such techniques are available.
I'm actually searching for such a system where we actually, without invading the animal, non-invasive procedure, if we can analyze these protein levels by, of course, tagging with some radioactive ligand or whatever it is. That will make the things much simpler. That is one thing what I was trying to find out. Maybe it is available abroad. I don't know. Here, it is available, as you said.
No, the in vivo images are available in India. In vivo imaging systems are available in India, but whether brain chemicals can be measured.
No, that's what I am talking. In vivo it is available, but specifically for this purpose. Anyway, that's one thing what I wanted to see. Second thing is, what is I just wanted to find out from you, in the absence of a challenger, like you said, monosodium glutamate challenge, and they are recovering their cognitive abilities or learning and memory aspects improved enormously. I'm sure that I think you did mention, but I was not. I did not catch it. In the normal animals, did it show your compound show much better enhancement in the water maze? Did it reduce the time to reach the platform?
Marginally, it was not very significant. Marginally,
Okay.
when you give the compound, we always see that at the compound treatment alone, there is
Okay.
some kind of an improvement in case of some of these compounds.
Okay. Okay.
We simply give the compound.
Yeah, that question I am asking because all we are talking about memantine improves the memory, Brahmi improves the memory. So you
Yeah.
give a person who is not learning very well some of these preparations made by Brahmi or whatever it is. So in a normal individual, not a challenged individual where his cognitive ability has come down drastically because of various reasons. So we can have a formulation for the normal human beings. Okay, you take this particular concoction, it will improve your memory greatly. That's why I'm asking whether
Right.
your drug improves the learning and memory and retention. All these things in the animal. That's what I said. You said it is marginally, not
Marginally, it was never significant. We did not see
Not-
such a, anyway, we were not really looking for that, but still, as part of the control, we would be doing that. So we find that it doesn't, by itself, it is not having any bad effect. And it is as good, or maybe there is a marginal, but not statistically significant improvement in some cases.
Yeah, it is. I think it will be quite interesting, no, sir, in the sense that whether it actually works in the normal, because challenging, making it is cognitive ability is impaired by challenging with some chemicals. Then we say that it recovers from that is one aspect. As it is, so many of us require, to improve our memory as we go old, or some of the students are not able to pick up, and trying to improve in normal human beings or the animals is another aspect. I know. I was just wondering whether such things can be thought of.
Yeah, I think in a normal animal or in a normal condition, we still are challenged as part of a daily stress. So some compound of this kind might be providing, could provide the daily wear and tear in the tissue, or in the brain, in the neurons, might be reduced by having some compound like this. So that is a possibility by which some enhancement in performance can be achieved.
Yeah,
that's
right.
my opinion.
Yeah, that's right. I mean, it's a very interesting talk, sir. You have worked very extensively in this area, and some of our students are also working. I mean, basically similar to the things like what you are doing. We are trying to analyze NMDA activity or acetylcholinesterase activity here. And when we did that, and of course with the in vivo studies using the behavioral studies like water maze or absolute maze, and trying to find out whether they retain the memory or get into the food or the reward quickly, and all those, basically measuring the latency time. So while we were doing, I was asking my students or my colleagues also whether in your you mentioned that in cancer or this image of the brain, you see the dopamine color in the coming in the red color, acetylcholine color in blue color, and all. I mean, giving a clear as control as compared to the control, the treated has got much more level of acetylcholine in the image. That is what we are looking for, and that's what I think I'm searching for now. Maybe actually, as you said, equipment may be very expensive. I don't know. I have no idea. That will directly give us what is the change which has taken in the CNS.
Correct.
Yeah, but your talk was quite. Your team has done a great work, sir. Thank you very much for elaborate presentation. And as you mentioned, I mean, some gentlemen had asked whether we can collaborate. I think we can work together, and some of the things. That's what I know. Well, either my people can visit you, or you can come here, and let us see whether we can form a group and take the things forward. Because the strength lies in collaborative work rather than working in isolation. That's how it is. Yeah. Thank you, sir.
Thank you.
Sir, one Mr. Prakash Patil wants to ask, what is the impact of tackling derivatives on calcium channel activity?
Tackling derivatives, I showed the data, if you remember, that they were causing inhibition of Nmda receptor activity. Nmda receptor calcium channel activity was inhibited as expected. I mean, that is how we, the compounds, hypothesis, the compounds were designed with that purpose. In the Nmda receptor bound tackling, additional derivatives were designed, additional groups were designed, and then they were synthesized. Many of them were actually inhibited Nmda receptor activity in the in vitro assay. So the calcium channel activity was measured with our assay. We do not, I don't know whether the person wants to know whether we measured the calcium level inside the cell. We did not do that with the tacrine derivatives. We have done that with other NMDR inhibitors, and we have shown that yes, the NMDA receptor induced calcium influx also can be blocked by this by NMDA receptor blockers. But we did not do that for this set of compounds, tacrine derivatives. So that is level of calcium directly, but we are looking at calcium induced events, and therefore calcium channel activity has been measured. It does block calcium channel activity.
Sir, our MPharm student, Ms. Apurva, wants to know what may be the implication of calcium channel blockers, which are patients, our elderly patients will be using calcium channel blockers as drugs for hypertension, angina, etc. So we were talking about the importance of calcium signaling in Alzheimer's disease. So what may be the implication of calcium channel blockers? So patients who are taking calcium.
Right, calcium channel is a very broad term. There are several different calcium channels, several different proteins, hundreds of proteins which have calcium channel activity. So under the bracket of umbrella of calcium channel, there are several proteins. So we would be designing inhibitors not for all the calcium channels. We would be designing for specific calcium channels. So calcium channel blockers against voltage-gated calcium channels are the ones which are used for hypertension, cardiac ailments. But what we have talked today are blockers against the brain calcium channels. Again, in the brain, glutamate mediated, glutamate activated calcium channel. That is what we have looked at. So these would not have, would not be acting on the calcium channels related to hypertension and cardiac function. Those are voltage-gated calcium channels. So the voltage-gated calcium channel blockers are used for angina and hypertension. So because voltage-gated calcium channel function is impaired in those conditions.
One last question from Omkar Mali again. What are the in vitro and in vivo models for normalizing beta amyloid protein?
For normalizing, what is it?
Yeah, normalizing beta amyloid. There will be accumulation of beta amyloid protein in various conditions. He's asking what are the in vitro and in vivo models for normalizing beta amyloid? And he meant by that.
No, in vitro assay is beta secretase activity assay, which I just mentioned. Other than that, neuronal cultures should be possible. I have to look into it. In cultured neurons, if you add A beta peptides, and then normally that will be toxic to the cells. But it may be possible to induce some amount of beta amyloid accumulation. And there are several in vivo Alzheimer's disease models. I have shown a model which is not an Alzheimer's disease model, which is a glutamatergic, hyperglutamatergic model. Glutamate-induced excitotoxic model is what I had shown. There are several Alzheimer's disease models where both pharmacological as well as genetic models in which plaques can be observed in the animal. Plaques should be possible. It should be possible to observe plaques in the brain.
But I think he is asking how to normalize it. Normalize, that's what we are all trying to normalize it, but not able to do it successfully for a period of time. The accumulation of the plaques, beta amyloid plaques, is one of the reasons. I mean, that is one of the important reasons for Alzheimer's disease. And we, for so long, we have been trying to, prevent the accumulation of the plaques. And that's what I think this research is all about, is going on. I don't know whether he asked about the model and whether he asked about what are the remedial steps are there to prevent the formation of the plaques. The prevention is miles away. I mean, still I think we'll have to work it out whether it can be.
Exactly. Right.
Dr. Nimi, before any, I just want to remind, before you sign off, I request you to take the screenshot
We have done, sir.
of this.
We have done, sir.
You have done, no?
We have
Okay,
done.
fine. Yeah.
Thank you very much, sir, Dr. Om Kumar, for the loads of information that you have given us. And we look forward in the future for more interactions or webinars and God willing collaborative work with you and your department, sir. Thank you very much.
Thank you. Thank you
Now
very
I
much.
request Dr. Prerna Shetty to deliver the vote of thanks.
I hope this seminar was an eventful one and has inspired the participants to take interest in the field of neurosciences. Now, I am privileged to propose the vote of thanks on the occasion of this webinar on multi-target directed ligands for neurological disorders conducted by Department of Pharmacology, Nitte Institute of Pharmaceutical Sciences, Nitte deemed to be University. I thank our Honorable Vice Chancellor, Nitte deemed to be University, Dr. Satish Kumar Bhandari, for giving opening remarks. I extend my heartfelt thanks to the resource person, Dr. Om Kumar, for taking his time from his busy schedule, accepting our invitation and delivering an excellent presentation and making this webinar interesting and informative. I would like to express my gratitude to our principal, Dr. C.S. Shastri, for welcoming the gathering. I am happy to express my thanks to Head of the Department of Pharmacology, Dr. Gururaj MP, my colleagues, and all participants from various institutions for attending this webinar. I also extend my thanks to Dr. Nimi Varghese for conducting and coordinating today's webinar. I extend my gratitude to all those who have directly or indirectly helped us in conducting this event. Last but not the least, I would like to thank Nitte deemed to be University for providing all the necessary facilities needed for conducting the webinar. Thank you, one and all.
Thank you.
Thank you all for finding time to join the webinar. Hope you had a wonderful learning experience. God bless you all.
So with your permission, I will leave the meeting. Thank you, Nimi.
Thank you.
Thank you, Om Kumar, sir. Thank you.
Thank you, sir. Thank you, Doctor. So,